It's great to be here. Welcome to the Plumb Auditorium in Christ's College, Cambridge. We're excited to be here. The second of our Whewell Centre Autumn Lecture Series. My name is Alistair McKittrick, and it is my pleasure to introduce and host this evening's event. The Whewell Centre takes its name from William Whewell, who was Master of Trinity College and was a contemporary of Charles Darwin. They walked and talked together on several occasions, and in fact, Darwin acknowledged the influence Whewell had on his own inductive methodology. When introducing a model of reproduction called pangenesis, he drew from Whewell's arguments to defend his speculative hypothesis, even though it was not yet a worked out full theory. So he writes, as Whewell, the historian of the inductive sciences, remarks, hypotheses may often be of service to science when they involve a certain portion of incompleteness and even of error. So this quotation in Charles Darwin's The Variation of Animals and Plants Under Domestication, published in 1868, which is 2 years after William Whewell died. The quotation was from volume 2 of William Whewell's book, The Philosophy of the Inductive Sciences. You can see it there on the right-hand side, first published in 1840. This connection between Darwin and Whewell is interesting for many historical reasons. First of all, Whewell's work to transform what counted as science from reliably true statements of fact into contested hypotheses supported by varying degrees of evidence, that was as significant and influential as was his coining of the term scientist itself. It enabled Darwin to defend the status of his hypothesis of natural selection using the terminology of an authority figure such as Whewell. This connection between methodology and authority cuts both ways. Intelligent design theorists, such as Stephen Meyer, have also drawn authority and support for their own scientific methodology by using the same induction approach as Charles Darwin and Charles Lyell themselves. That is to say, what is good for the Darwinian goose is good for the intelligent design gander. And William Whewell is behind them both, intellectually speaking. Although more recent methodological discussions tend to appeal more to the late Cambridge philosopher Peter Lipton's work on inference to the best explanation. This mention of Charles Darwin is very relevant to this evening's talks on the origin of life and the information in the cell. It was Darwin's musing on the chemical basis for abiogenesis that gave comfort to his whole project of providing a materialist explanation for the origin of species. In a letter to Joseph Hooker, 1871, Darwin wrote, "The original spark of life may have begun in a warm little pond with all sorts of ammonia and phosphoric salts, lights, heat, electricity, etc. present, so that a protein compound was chemically formed ready to undergo still more complex changes." Well, this hypothetical conjecture is the subject of our lectures tonight. So without further ado, I'd like to introduce Professor James Tour. He is a synthetic chemist. After spending 11 years on the faculty of the Department of Chemistry and Biochemistry at the University of South Carolina, he joined the Center for Nanoscale Science and Technology at Rice University in 1999, where he is presently the T&T and F.W. Chow Professor of Chemistry, Professor of Computer Science, and Professor of Materials Science and Nanoengineering. And it was at this point that I was going to read out a few of your areas of expertise, but it would simply take us too long. The one that I especially want to highlight was your interest in racing nano cars. That seems pretty cool. Okay, Dr. Tour's talk this evening is entitled, Are Prebiotic Simulation Experiments Simulating the Origin of Life? Please welcome Dr. James Tour.
So this is, what I'm showing you here is just an overview of many of the areas that we are working in. And the reason I show this is not to go through them, but to tell you that I'm a practicing scientist. So it's not like I do this, I talk about science as a hobby. This is what I do every day, and this is what I do all the time. And when I talk about science at the dinner table at home, my wife gets up and walks away. So she's had her fill of this. This is what I work on. This is a list of the companies that have come out of my laboratory over the past 10 years. And so it shows you that we work on things that are not just pie in the sky and not just maybe benefiting the world 50 years from now, but we work on things that are practical and can be practical immediately. Now, not everything is practical immediately, but many things are. And so 3 of these have now become public companies. And again, just to show you that I think a lot about the practical issues in science. So let's talk about abiogenesis. There's definitions of this, and abiogenesis is the origin of life from non-living matter. To construct any convincing theory of abiogenesis, we must take into account the conditions of the Earth about 4 billion years ago. That is not my definition. That is a textbook definition for abiogenesis. And that's what it says. So you see what we have to consider according to the textbooks. Now, prebiotically relevant means that we're restricted to materials, procedures, and conditions that might have been available on an early Earth. What was the Earth like? And so we didn't have chemical companies around. We didn't have chemical plants. We couldn't just order chemicals from the manufacturer. And you only had very basic small molecules. You don't have all the molecules that biology has supplied to us on this Earth. You have small molecules like carbon dioxide, nitrogen, ammonia, maybe some hydrogen, but very small molecules. Now, nobody was present at life's origin, so we'll never know how life originated. But that's not what we're seeking to answer. What we're seeking is an experimentally verifiable hypothesis as to how life might have originated. That's all we've got to do. Just tell us, give us a proposal on how life might have originated such that you can begin to mimic this in your own laboratory and bear something out for that hypothesis. None of this, it might have happened in a small pond somewhere. All right, so what are the characteristics of life? So this is not what is life. What is life is something that's probably explained to us better in the Bible. But the characteristics of life, science can give us, and it's responsiveness to the environment, growth and change, ability to reproduce, have a metabolism and breathe, maintain homeostasis, being made of cells and passing traits on to offspring. Now, homeostasis is the steady internal physical and chemical condition. Now again, now what's happening, the reason I underscore this is the definition of the characteristics of life. This has been around for a long time. What people are doing now is they're trying to redefine this into something that they can make happen. And usually they'll take one characteristic or 2 characteristics and say, hey, we kind of have that now, so we've made life. No, you can't redefine this thing. This has been around a long time. So we can't redefine life into something that we can make in our own laboratory that wouldn't do all of this. So I'm not here to talk about God of the gaps. This is not what I do. I've been accused many times of being a person of God of the gaps. And I always contest with them, and I'd say to the people who accuse me of this, show me the video or show me the document that I've written where I talked about we just don't know, therefore God must have done it. And they can never find it because I never speak about that. And the reason I don't speak about that is that we just don't know what the future will hold. So if you asked somebody in the year 1600, when some of these buildings around here were built, would we ever be able to put people on the moon and bring them back? And they'd be like, I don't know what you're talking about. We don't have flight, let alone space flight, let alone traversing that far. So they wouldn't have had a construct in their mind to allow them to frame even such a question. And what we're talking about here is that because I don't know what the future is going to hold, maybe in 500 years or 1,000 years, people will certainly know a lot more than we know now. So I could never say, if we don't know an answer, therefore God. I would never use that as an argument. All right, so molecules are the language of living systems, in that everything is constructed of molecules. Every living system, you have to come up with these 4 classes of molecules. You have to have polysaccharides, polypeptides, polynucleotides, and lipids. But each of those is made from a smaller building block. And the smaller building block in this case is, for polysaccharides, it's monomeric sugars. You have to have the amino acids, you have to have the nucleotides, and you have to have glycerol. Each of these is a huge problem. Everywhere where you see a red arrow, that's a stereogenic center. You have to be able to control that. That's the whole chirality issue, left-handed, right-handed. Very hard to make. There is no prebiotic route to monomeric sugars. There is a prebiotic route to amino acids, that is the famous Miller-Urey experiment. But it's interesting, those mixtures that come in the Miller-Urey experiment would be unusable. Yes, you can see amino acids forming, but it would be unusable. And it would be unusable because it's such a vast mixture. The majority of the amino acids that are there are glycine and then alanine. And those two swamp the system so badly that there's much less of these other ones, and it wouldn't be usable because the mixtures are too great. No one has ever taken anything that's come out of the Miller-Urey type experiment. Many people have reproduced it and be able to use that mixture. So it was unusable. Nucleotides, you have to first have the sugar, then you have to have a phosphate, and then you have to have a base. Nobody's ever made a nucleotide in a prebiotic sense, and nobody's ever been able to take glycerol and make a lipid because you have to be able to distinguish the two OH groups on the end. Those are in what are called the enantiotopic centers. But this is the language of living systems. This is what we've got to deal with. But molecules don't care about life. Never have they moved toward life. Molecules have never been known to move toward life except in an organism or a living system already. Organisms can move molecules to keep themselves operating, but it doesn't happen outside of an organism or a human being pushing them toward a living system. They don't know to move in there. They have no brain. They don't know to go toward this. Now, has the public been misled on the origin of life claims? So the public has been asked, under simulations of Earth's early atmosphere, scientists have mixed molecules together in a laboratory to produce complex life forms such as frogs. 1/3 of the general public believes that is true. And the number 2 question, under early simulations of Earth's atmosphere, scientists have mixed molecules together in the laboratory to produce simple single-cell life forms such as bacteria. 2/3 of the people in the world think that that's true. That's really odd because both of these are false. And it's not only that they're false, it's emphatically false. They're nowhere close. And who were these people? I mean, well, the average age was 38, half of them male, half female, and 80% hold college degrees of some type, from associate's degree in college to PhDs. So this is the population we're talking about. 80% of the people think that these two things are true. Now, how could that be? Well, there's been mixed messaging from the community itself. And so, for example, Professor Jack Szostak, who has a Nobel Prize, he was at Harvard about 3 years ago, he moved to the University of Chicago. But he said that he would have life in the lab in 3 to 5 years, and that was in 2014. Guess what? He missed his deadline, all right? And it's quoting him here. You heard it here first, announced World Science Festival moderator and astrophysicist Mario Lidio as Harvard biologist Nobel Prize laureate Jack Szostak told Search for Life gathering of 50 on Saturday afternoon in New York that he expected to make life in the lab in 3 to 5 years. His colleague, Dimitar Seselov, who's also at Harvard, he said, well, probably closer to 5 years than 3. So that's the message that went out from these people. Well, remember, that was in 2014. In 2021, at a lecture at the University of Chicago, a virtual lecture, so it's online, he says that he has not even made RNA yet. He says, and basically RNA is really unstable. It's really hard to get this thing. It's tough. And so he confesses he hasn't even yet made RNA from a prebiotically relevant root. Well, RNA, even if you have an RNA, from RNA to life is a huge, huge task. Nobody knows how to do that. He hasn't even made RNA. But why is it that 7 years earlier, he was saying he'll have life in the lab in 3 to 5 years? He hadn't even made RNA yet. This is the mixed messaging that goes to a lay audience versus to a scientific audience. Steve Benner, formerly at Harvard and now director of the Foundation for Applied Molecular Evolution, he said to a YouTuber, so it's all recorded, he says, I suppose many of the big paradoxes in Origin of Life have been solved. So he said most of the paradoxes have been solved. This is what he told this YouTuber. I don't think a single paradox in the Origin of Life has been solved. Zero. None of them have been solved. And he says most of them have been solved. Well, here's what he said at a professional meeting in 2019. He said, chemistry is actually hard to get to work. The molecules precipitate, the molecules hydrolyze, the molecules decompose, and so it's very much a constraint that you have to deal with. It's one goddamn problem after another. Now that's true. That's true. That's what he told the scientific audience. That's the truth. Why would he say most of the paradoxes have been solved? I confronted him at a meeting in Israel a few years back, and as soon as he came to the gathering, I saw him coming out of the bus, and I went right to him. And I said, tell me, what paradoxes have been solved? Can you tell me? He couldn't tell me. He wouldn't say anything. And we talked, and he told me nothing. He told me nothing. There's nothing there. Professor Cronin, he said he posted, this was posted September 9th, 2011. He said, he gave a TED talk, and he says, what I'm going to try to do in the next 15 minutes or so is tell you about an idea of how we're going to make matter come alive. Okay, that's interesting. I want to hear that. 15 minutes. You can explain that to me. Hey. And at the end of his talk, Chris Anderson, the moderator, said, just a quick question on timeline. You believe you're going to be successful in this project? When? When? Here's what he said, hopefully in the next 2 years. That was in 2011. Guess what? He missed his deadline. He's nowhere close. Nowhere close. This is the problem. This is Jeremy England, who was an MIT physicist. It says here, an MIT physicist has proposed the provocative idea that life exists because of the law of increasing entropy, drives matter to acquire lifelike physical properties. Let me get this. Increasing entropy? Now, life has very low entropy, high energy, low entropy. We're ordered. Molecules in us are not just flying all over the place. But he says increasing entropy drives matter to acquire lifelike properties. And he said in this article, you start with a random clump of atoms, and if you shine light on it for long enough, it should not be so surprising that you get a plant. And so I wrote an article and I said, people have already tried this experiment with all sorts of wavelengths of light. They've shown that light upon atoms of all different types, and life has never sprung up. Never. So I would be very surprised if that happens. He says you shouldn't be surprised. And this is the problem that happens when people say these sort of things. So, embellishment by the press. So then it's taken up by the press even further. So like this came out just August 30th, just a couple of months ago. Scientists say they may have just figured out the origin of life. They figured it out. I didn't know. I didn't know. And it comes down to some very simple chemistry. So I looked up the article. Well, this particular article, it says in this article, the results he added show how RNA might have first come to control protein synthesis. Well, Nick Lane, an Origin of Life chemist at UCL, who wasn't involved in the study, further cautioned to science that the amino acid chains being produced are random and chaotic, unlike the orderly arrangement produced by ribosomes. They still have not cracked that problem, he told the magazine. He was being really gracious. He said the order of attachment, it's still kind of random. Everything in the article was crazy. But here's what it says at the end of that article. In response to Nick Lane says they really haven't cracked the problem of ordering. He says, but give the chemicals billions of years to bounce around and anything can happen. So that solves it. If we just waited longer, that would have solved it. That's the messaging that comes out. So I looked up the actual article, and it's called Thioester-Mediated RNA Amino-Acylation and Peptidyl RNA Synthesis in Water. And so what this article is about, it has nothing to do with forming life. And so you look it up and you see what he started. He purchased highly purified enantiopure amino acids with Bach protection. Bach protection means there's a human group on there, a human designed group attached to the amino acids. It's not like the amino acids. First of all, it's chirally pure, so it's enantiopure, and it's already protected in a protected form. The amino acid thiols were prepared using purely synthetic methods on Bach protected amino acids, HOBT, which is a human designed activation group, diethylamine, which is a human design based. This DIC is a coupling agent, DIPA is a base, and so you use dichloromethane as a solvent, and you have, again, these amino acids are protected using 4 molar HCl to remove the Bach. Everything is synthetic behind everything he made. Either he bought it or he had to use synthetic methods. Then he started with RNA oligomers that he purchased. And where do these oligomers come from? They come from, either you synthesize these in a laboratory or you take them from a naturally living system that's already alive. And his yields with many of these systems was about 10%. Now you have to have 99.99% coupling yield in order to get any reasonable length of a polymer. So he was only making dimers, trimers, or the biggest was a tetramer. That's 4 units. You have to have hundreds of units long for these to work, and under highly specified conditions. In other words, there's nothing in this. What happens is you read this article, and it cries out to you, life did not happen this way. All of their experiments cry out the same thing. I look at the same data that they say is making life very simple. This cries out to me, life could not have happened this way, because in all of that you set up, sometimes you're only getting 10% yield. If you don't have 99.99% at each coupling step, you're toast. It's over. You can't do this in what's called a condensation polymerization. So that leads to textbooks being corrupted. So here's a sampling of 41 textbooks suggesting the primordial soup. That same thing that Darwin was talking about in that letter that was just put up there, this primordial soup model. This is not just taught in college, it's taught even in graduate schools. Upper level graduate school textbooks still talk about the primordial soup model, that you had some pond with small molecules floating around in it, lightning strikes, the molecules formed more sophisticated structures, those came together, formed all these 4 classes of polymers, those came together, formed a cell, and those cells formed multicellular organisms that then formed these structures that came slithering out of the pond. That is the primordial soup model. That primordial soup model, I was wondering where it had come from. John Lennox told me, since he had taken classes from C.S. Lewis, C.S. Lewis said it came from Babylon. The Babylonians had a pond that they said life came out of. They said their gods also came out of that same pond. And this is where the primordial soup model came from, that Darwin was talking about, that is still in our graduate level textbooks today. That's the problem. So what's the true state of the origin of life research? Well, nobody has ever shown a method to make the enantiopure versions of carbohydrates, these amino acids, nucleotides, or lipids. Nobody's ever made these basic building blocks of the building blocks. Nobody's ever shown. There is no method. And in fact, the monomeric carbohydrates, there was a reaction, there's something called the Formose reaction, and it was thought that the Formose reaction is an autocatalytic reaction. Well, I looked at the data of the Formose reaction years ago, and I said, there's nothing here. He hasn't shown me anything. I don't see, even in their own data, I don't see ribose, which is really what you're going to need for making the nucleotides. I don't see ribose there. And the NMRs don't look very clean to me. So just recently, just this year, Ramanand Krishnamurthy from Scripps Institute went back and visited that reaction. He's an origin of life researcher. His group tried over and over again. They couldn't make ribose. That foremost reaction doesn't make these sugars. It makes other things, doesn't make these sugars. And it's not autocatalytic. The one reaction, they had one reaction in all of origin of life chemistry. One reaction that they said was autocatalytic, meaning that it forms and then it goes faster and faster and gives you the product. It turns out that that one that was claimed to be autocatalytic is actually not autocatalytic. So the one that you had, that you said autocatalysis is the basis behind all of this origin of life, you had one reaction, and that one turns out not to even be. It's a real problem. Their whole area is just crumbling. It is really crumbling. It is only alive in their own minds. Nobody's ever shown life could form with lower enantiomeric excess. Some have said, well, having high enantiomeric excess, having these chiral elements came as an afterthought in life, that evolution made that happen later on. Well, you had to have had it beforehand. There's work in this area called chiral-induced spin selectivity, which says the reason why you and I do all these calculations and our brains don't just burn up. If you put your hand on a computer chip, it would get burned terribly. Your computer chip gets very hot. Our brains only put out less than 10 watts. And there's a reason for that. And because you have these molecules with these electrons, and these electrons in these molecules can be either spin up or spin down. And when we do chemistry, we don't control for that. We use them both. And that's why our yields generally might be 90% on a good day. But in biology, the yields are 99.999%. Why is that? And it's because our bodies have the ability to choose out selectively one direction of the electron over the other. There's one direction over the other. They're what are called spin valves. They allow one and not the other. And because of that, you don't get backscattering. And because you don't get backscattering, you don't generate the heat. And this is why we don't burn up. And all our cells don't burn up. It's really remarkable. We didn't know about this 25 years ago. So this is a newer discovery which tells us life never could have formed in the first place if it didn't solve this. And to solve this, you had to have solved the chirality issue. You had to get the one-handedness and not the other, which is hard to do in a laboratory. How that happened on an early Earth, nobody knows. People have talked about chiral surfaces giving you the induction. People have studied chiral surfaces for 40 years. Nobody has seen these levels of induction that you need to see. In human intelligence put behind generating the right chiral surfaces, it's never been sufficient. So nobody's ever shown that the mixtures found in meteorites or interstellar space could be useful. And I'm not the only one who says this. John Sutherland, who's an origin of life chemist, he says, I don't think meteorites make the right sort of mixture, but we need to have more constrained chemistry to actually make the right sort of mixtures. Now, John Sutherland is an origin of life chemist not far from here, but they will never engage with me. You'd think that they want to come to a talk like this and engage with me and talk. I have invited any one of these guys to come on my channel and dialogue with me. And if you're afraid to come on my channel, I've said, I write to them if they have a paper come out, hey, we'll go on a neutral channel, or I'll go on your channel. Let's discuss this thing. I was asked to be on a very popular program on the internet and to have a debate or a discussion with any origin of life chemist. An origin of life chemist says it's going to have to be someone who works in origin of life, or an organic chemist, or a biochemist. And he said, give me the names of people I can invite. This is a broad program that many of you, I'm sure, have seen. And I gave him the name of 10 origin of life researchers. After a few months, he contacted me. He says, would you come on the program? I said, yeah, I'll come on the program. Could you get anybody to come? He says, nobody will come. I gave him another 15 names of people. He's never come back to me. Why will nobody engage with me? That in itself is telling, because no response is an answer in itself. They will not engage with me to talk on this subject, even the experts in the field. Nobody's solved the mass transfer problem. This is a huge problem. How much material, if you start going from small molecules, these are complex molecules compared to what early Earth had. But if you go, how much do you need to get to a living system? What happens in the lab, you start working. If each yield is 50% yield, then you have 50%, 25%, 12.5%. 6.25%, and you run out of starting material. So what you do is you optimize the reactions, and then you go back and you look at your laboratory notebook, and you go back and you start building more molecules according to the ways that you had figured out before. You can go back to your laboratory notebook and you keep bringing material from the rear. You can't do that in this case because nature never kept a laboratory notebook. Doesn't know what nature did. So it might be going on for a few million years, and then it needs to go back and make more starting material, but it can't because it doesn't know how to made it in the first place. Nature doesn't keep a laboratory notebook. No person has ever addressed the mass transfer problem. This stops everything. Nobody's chemistry can ever work without solving this problem. Half of the amino acids, everything you see in a red circle, half of the amino acids have a reactive side chain, and a few of them are somewhat reactive. So you have this reactive side chain. If you can't block that, then you can't do this type of chemistry. Remember I told you that he bought Bach-protected amino acids. Why Bach-protected? Because you protect them. In his case, he was protecting a certain end over another end. You need to be able to distinguish this. This is how human beings do synthesis. You had to be able to, nobody's addressed how you do synthesis of polypeptides with these end groups not Bach protected. It's a big problem. So what Pauner did in that article that I cited, he used an RNA template. He already started with RNA. So he started with nature's construct. So that's how he was able to do this. So I've challenged them. I said, if you even make a dipeptide to hook together just 2 amino acids, here's 2 amino acids. Can you hook these together in a reasonable yield of, say, 90%? Because remember, you're going to have to get 99.999% for this to be useful. Just do it in 90%. I said, if you solve this, I challenged on the internet, all the origin of life chemists. If you solve this, I'll stop talking about origin of life. I won't do any more meetings like this, and I'll pull down everything on my YouTube channel. I shared this with Steve, that I'm going to ask. He says, Jim, he was very worried for me. I said, don't worry. I'm an organic chemist. I can write an exam that nobody can solve. I know how to do this sort of thing. And nobody solved it. And I would let them solve it. And I also challenged them on this problem. Nobody's ever solved the nucleotide polymerization problem, because what you get is instead of this 3-5 linkage, you see here there's a 3' to 5' linkage. This is a 2' to 5' linkage. If you can't prevent this from happening and the branching from happening, you can't make a polynucleotide. You can't make RNA. I said, you just solve this problem and we'll go on with it. And I said, here's the challenge. And if you can make me a 200-mer with less than 2% of the 2-5 linkage, less than 2% of the branch, nowhere close, nobody came forward, it can't be solved. I said, you solve just this one, and I'll stop talking about it. They can't solve this. Nobody solved the carbohydrate problem. You can take 6 carbohydrates. You take this glucose, 6 of them. How can 6 units of the same units be hooked together? A. How many different ways can they be hooked together? Well, of these carbohydrates, you can hook these together over 1 trillion ways. Why? Because each one of these OHs is an attach point. So you have all these different OHs, plus these rings can be 5-membered rings or 6-membered ring, or they can be the open form. This is a huge problem. Every sickness that we have as human beings, every sickness harkens back to an associated carbohydrate linkage problem. If you don't get this thing right, the cell dies. Nobody has any idea how this was done in a prebiotic fashion. To do this synthetically in the lab is very hard. Carbohydrate chemists are the best chemists because this is such a hard job. But you're using human-made designed methods to do this, and it's arduous, difficult chemistry. Nobody's ever solved this. Nature does it no problem with its enzymes. But remember, they have to do this prebiotic before natural systems are around. And I said, just prepare the diet. Just hook 2 together cleanly. In over 90%. Just 2. You don't have to make the problem. Just 2. Nobody came forward. Nobody could answer this question. The other problem is that all of these polymers decompose. So they have a half-life. But half-life means that you're starting with a large number of them. If you happen, some people will say, well, you happen to get an enzyme that had the right amino acid sequence. And over the billions of years, we just read in that article, with the billions of years kicking around, No telling what might happen. Well, I can tell you what's going to happen. Within a matter of days, that molecule doesn't exist anymore. It undergoes hydrolysis. So when you look at RNA of a 600-mer, it will last about 4 hours. You have to take the half-life and you have to convert it now to a probability, because half-life says you get half the amount of material after time X. But you don't have, when you only have one, you don't have you can't just take half of the material. There's only one. So it only lasts 4 hours. Of a polypeptide, it lasts about 13 days. That's it. So things don't have billions of years to slam around with each other. If for some reason you got something, it decomposes. It decomposes. And it never could have polymerized in the first place if you have other molecules around confounding this. So nobody can explain the protein folding problem. You have a protein. How does it fold up into the shape it needs? Most proteins need chaperones. They need other proteins to help them to fold up. And it's like taking you've seen these hammocks that are made out of a rope that's kind of woven, and you get this hammock. Imagine if you took that rope and you just threw it on the ground and you said, I'm just going to let the wind and the rain weave me a hammock. You can wait billions of years. It's not going to happen. It's not going to happen. That's what you have to solve. And in fact, the estimate is here by these authors is that if you had a single protein that was about 100 residues long, which is a normal size, not a particularly large protein, it said it would give you 10 to the 95 possibilities. 10 to the 95 is a very big number. He even says that even a small protein that initiated folding by the random search at that time of the Big Bang would still be thrashing about today. If it's over 10 to the 40th, there's not enough time, even in a 14.8 billion year universe. All right, so that's a simple problem. The harder problem is the non-covalent molecular interaction problems, which is called the Leventhal 2.0 problem. Just in a single yeast cell, which is a very simple cell, the number of ways that just the proteins can align with each other, can interact with each other is 10 to the 79 billion. That is a 1 with 79 billion zeros after it. The number of elemental particles in the universe is 10 to the 90. 10 to the 90, a 1 with 90 zeros. That's the number of elemental particles in the universe. This is 10 to the 79 billion. There's no way, we have no idea. You go to numbers like this, it's crazy. It's just crazy. In a billion universes, there wouldn't be enough time for this. And so in every cell in our body, there's non-covalent interactions of how molecules align. So when cells divide, they take that information and they start moving it to each side of the cell, and then the cell next down in the front, and now you have two cells that have that inherent information. But where did that come from Luca, the last universal common ancestor? Nobody knows. How do they address this problem, the origin of life researchers? They don't. They don't. It's unaddressed. Nobody has solved the code problem. You need to have some code here, some orientation of molecules in order to give you a code. Just a random box of letters gives you no information. You could shake that up all day. You might get a simple 3-letter word to line up, but you don't get any inherent information that's suitable for life. Nobody knows where the code problem came from, how that was solved. Now, nobody's ever made the higher order structures. To build a cell, you first have to build all of these different pieces: DNA replication, repair, restriction, and modification, basic transcription machinery, and all. You just go down this list. This is the simplest cells. People will argue, oh, Tour, you're just talking about complex cells. No, this is the simplest cell. So you can look at simple cells that we have today. They're not much more complex than the simplest cells we have found in the fossil record. So the simplicity of the cell has not changed when you talk about our simplest cells. Moreover, bioengineers have calculated back, and they've shown the simplest a cell could possibly be. Here's the simplest it could be. And it's so simple, it can't make any of its own amino acids. You're going to have to supply that all from the outside. This is a very simple cell. But you need all of these different subcomponents. How many of these subcomponents have been made by origin of life researchers? None. Not a single one. So how can we be near understanding life? And these folks in the origin of life community still keep embellishing this and then speaking to the press and letting these articles go out with this kind of nonsense. And then it gets into our textbooks. And this is what we learn and our children learn. And now, it doesn't matter what hypothesis you come up with, RNA world hypothesis, protein world, metabolism first, lipid world, hydrothermal vent hypothesis, and then some others that have fallen by the wayside. And as Steve Meyer says, they keep recycling these. Every few years, they bring another one back up to the top. None of them work. All of them fail for every one of the chemical reasons that I've given you. And you say, wow, Tor, you really thought of all the chemical reasons. No, I am just presenting 5 of 5,000 chemical reasons that I can present why all of these don't work. So what's needed to make a simple cell? You have to first make these molecules at the bottom. And so nobody's ever made those. Okay, I will provide those to you. So say I provided those to you. Go ahead, just make the polymers. Nobody's been able to polymerize these with any sort of control. Nobody knows how to do this. They can't make the polymers. If they don't have the control, if you have the polymers, you already have the code for life. The code is embedded in the string. So the basic ones are the letters, but if you have these in the exact order. So I'll give you those as well. So in other words, I'll give you those as well. What if I provided to you the polymers? Because we can take a cell and we can deconstruct a cell. And you have all the polymers there. So then I give these back to you, which already has the code in it. Can you build a cell? Go ahead, can you do this? So assuming you had access to all parts in your laboratory. So I take a cell, I deconstruct it, and I put the polysaccharides in one bottle. I put the lipids in another bottle. I put the nucleotides in another bottle. And I put the proteins, these polypeptides in another bottle. And then all the other components in different bottles. And you're not in a hole in the ground. You're not in a hydrothermal vent. You're in your modern laboratory. And you can get help from anyone you need. You know, call the helpline. Get help from... Can you just put the cell back together? There is not a scientist on Earth that would say that they can do that. Because we're so far... They can't even put Humpty Dumpty back together again. It can't be done. So we're just clueless on this. So how about the resurrection of a cell? Because a resurrection must be easier than a de novo synthesis. So just take a cell that's already just died. Just define for me what we just lost. That's a hard problem. What did we just lose when we went from a living cell to now a dead cell? It's hard to know. I mean, there was so much stuff going on. What just happened? It's hard to explain. But now bring it back to life, because I'm telling you, a resurrection's got to be easier than trying to pull all the molecules back together. Nobody can take a dead cell and bring it back to life. I mean, this is how far we are. We can't even resurrect a cell. So has science explained the origin of life? It's not even close. Scientists might explain it someday, but that day is far from today because we're getting further from the solution due to our increasing realization of the complexity of a cell. It's the moving goalpost. So say you say, here's the target, and here's where we're moving along. And you say, well, we discovered this, and so we're a little bit closer. Yeah, okay, you went a nanometer closer. But in that time period, this has gone 20 miles further out. Why? Because we understand the complexity of the cell. It's not that the cell has evolved into something different. It's just that 25 years ago, we learned that we had to solve chiral-induced spin selectivity. You're like, whoa, I got to solve that? I mean, that's another massive problem. And every year, we're learning more about what cells do, the complex layers of signaling they have, even within a cell. And so the target moves further away much faster. That's why in 1953, with the Miller-Urey experiment, that's why they thought you had, just from here, you had the understanding of how information is stored in DNA. Came right from this university in 1953. The same year, Miller and Urey came up with amino acids. They thought they were going to solve the origin of life. Very soon. But now, 75 years later, we've not solved this thing. Why is that? It's because the target has moved much further away than it was in 1953. In 1953, it looked doable. Now it looks like we have no idea. So my appeal to origin of life research is, if we're beset with so many barriers, regardless of the model, why not join me in sharing the obvious with the press and the lay public that we have no idea how life is first started. And it is obvious that life can be made from inanimate materials. So in other people say, you know, can life be made? We are a testament, we are evidence of that. Life can be made from inanimate materials. So there's nothing profound in that assertion. You know, obviously, there was a time when there was no life, and then there was life. So matter, whether it be the clay of the ground, the matter was turned into life, regardless of your model. So we already know that can be done. We are living examples of that, and it doesn't matter. If you want to take panspermia, where you have aliens placing it here, or it came here in meteorites, that just begs the question, because we're talking about the origin of first life. How did life first get here? So the problem is that any materialistic model that we invoke is far from having an experimentally verified hypothesis as to how life might have originated. All proposals fall far short. We can't even solve the dipeptide or the disaccharide problem with our models, just hooking two molecules together. So what are more relevant targets? What are the types of things we might want to be looking at? You address the mass transfer problem. How do you run out of material unless you can show me reactions that are 100% yield at each step? Solve that one. Come up with the reactions that are 100% yield at each step, then you don't run out of mass. The Leventhal paradox, the protein folding, and the interactome problem. Address that one. Try to figure that one out. How does that get solved? The making of a certain dipeptides or disaccharides. Solve those. These will move us along. The formation of RNA without the 2', 5' linkages, the chemoselectivity problem. A method to remove human intervention. In other words, everything you do in your lab has massive human intervention. Now try the same thing without humans. Just walk into the lab and go, something great going to happen today. And just wait for it to happen. Why doesn't it happen? Remove the human intervention. The secret to synthesis that can repeatedly survive impure mixtures or solve the separation problem. What happens is it gums up the works. You can't do chemistry with impure mixtures because those other impurities get in the way. They react instead of what you want. Or show that you can separate these. How do you do the separations? Nobody's ever solved that. Solve the relay synthesis problem. So they'll buy something from a chemical manufacturer and they'll assert it right here. Hey, I got this step to go. Just like Powner did. But you took it in an already advanced state and you did exactly what biology does, but you already took everything in this advanced state. That's the relay synthesis problem. Solve the condensation polymerization where the free energy is positive. All of these polymerizations I've showed you, it's called free energy is positive, meaning that they're unfavorable. They go back to the starting material. That's what's favored. Now, how do we do this in the laboratory? We set it up. We set it up in the laboratory, which what's called coupling agents that couple these. But without those, you have to solve this free energy problem another way. Nobody's ever been able to do that. The polymer stability problem, that these things only last hours or up to days in water and then decompose. We've got to solve this problem. The origin of specified information, the code. Nobody's ever solved that. Address that one. That would move us along. And then the final assembly. How would you assemble things? How would you assemble things if you had all the pieces? You solve that one, then that would be a great advance. And the final thing, resurrection. Just bring a cell back to life. That would be a good target. Take a dead cell and bring it back to life. If you can do that, that would move us along this road. So there are things that you could do that would be useful. So are prebiotic simulation experiments simulating the origin of life? No. They cry out life could not have occurred this way. It didn't happen this way. This is exactly what all of their experiments show. It didn't happen this way. So, and I have a personal invitation. If anybody here does not believe in the physical resurrection of Jesus Christ, you send me an email to tour@drjamestour.org. We'll get together and we'll talk about that. And I think you'll come out believing after an hour. All right? Let me give you some evidence for that. Here's how you can connect with me. I have things going on all of these social media platforms. And if you need to write to me about the resurrection issues, you just send it right there. Thank you.
If you do not believe in the physical resurrection of Jesus Christ, send me an email, tour@drjamestour.org, and we will get together, and I will share with you about why I embrace the resurrection of Jesus.
Excellent lecture from Dr. Tour. Thank you, James, for coming all this way. Some of you may not know just exactly how accomplished he is. I once introduced him by mentioning that he had 600 peer-reviewed papers, and one of his assistants reached out to let me know that it was actually over 1,000. So this is one of the world's really top scientists, and it's not surprising to me that people working in origin of life are not keen to engage him on the plausibility of the chemistry that they're citing. I had an experience like this myself. I did my PhD here on origin of life biology within the Department of History and Philosophy of Science. My supervisor had written a definitive history of origin of life research, and she came back from one of the international societies for the study of the origin of life, the ISOL meetings, and she said we had a tutorial, and she said, Steve, it saddens me to say this, but our field has reached a point of impasse. I even feel like we're getting to where a lot of people are crossing over the line into being a little bit like quacks and cranks. I said, what do you mean? She said, well, at the meeting, it's clear that everyone knows that everyone else's idea won't work. They're models, but they won't admit it about their own. And she said, that's kind of the definition of someone who's crossed that line. And that was 1989. As Dr. Tour has shown, there's not been a lot of progress since. Now, my supervisor in question was a woman named Harmke Kaminga, and she had an observation that I think helps explain why there has been so little progress. And Dr. Tour alluded to it at the end of his talk, and that is that she said at the heart of the origin of life problem lies a fundamental question. What is it that we're trying to explain the origin of? And as we've learned as we've learned more and more about the complexity of life, the problem has gotten more and more difficult. It's receded further and further away from anything resembling a solution. And so it also explains another mystery. It's kind of an historical mystery, because whereas Darwin did speculate about life beginning in a warm little pond, he was also quite clear that we didn't really have an answer to the origin of life. And at the same time, he was very adamant that he had explained away all evidence of design in life. There's a famous quote from one of his letters. He says, there seems to be no more design in the variability of organic beings and in the action of natural selection than in the course in which the wind blows. And this was actually very, if you think back on that period when Darwin is formulating his ideas about the origin of species, he's placing great emphasis on the creative power of his mechanism, natural selection. And he proposes it as a mechanism that can effectively do what intelligent breeders can do. It becomes a kind of designer substitute mechanism. He imputes to it so much creative power that he says it's not necessary to invoke intelligence at any point along the way in the history of life. And so modern textbooks report this same insight. They say it was Darwin's greatest accomplishment to show that the directive organization of living beings can be explained as the result of a natural process, natural selection, without any need to resort to a creator or any other external agent. So you've got this denial of design, a sort of comprehensive denial of design in anything that we could look at in the biological world, and yet, at the same time, Darwin is very clear. He says, it's mere rubbish thinking. This is a letter also to Hooker, I think. He says, it's mere rubbish thinking at present to say anything about the origin of life. One might as well think about the origin of matter itself. In other words, we're clueless. We don't know. And yet he also, at the same time, is affirming we have completely explained away every appearance of design because we have this mechanism that can produce the appearance of design without being designed or guided in any way. And that mechanism is the mechanism of natural selection. So how do you reconcile these two things? It's a little bit of an historical puzzle. Well, again, I think my supervisor's comment is illuminating because she's saying, well, the key thing is our understanding of life itself. And in the 1870s, in the immediate wake of Darwin's publication of The Origin, it was thought that life itself was very, very simple. This is a quote from one of the great scientists of that era, Thomas Henry Huxley. He said, the cell is a simple homogeneous globule of undifferentiated protoplasm. His counterpart on the continent, Ernst Haeckel, said something very similar. I love this because these were the great scientists of the day, and this looks so foolish to us now. We feel so much smarter than they to look at a quote like this. But they did not have the benefit of everything that has been discovered since. And so in the 1860s and the 1870s, Darwin could say, on the one hand, we've explained away all evidence of design. Every appearance of design in living organisms has been explained away because we have an undirected mechanism that can produce the appearance of design without itself being guided in any way. And at the same time say we don't yet have an explanation for the origin of life. But no one was worried about it then because they thought life was very simple. They thought it was composed of a simple substance they called protoplasm, which they conceived of as a kind of simple chemical goo or jelly. And if you think of life that way, you could imagine that a few simple reactions would move you from very simple molecules to life, because all you had to do was get this substance that was the essence of life. Now, of course, we've learned a lot about life since then and about what makes it work. And so that raises the question, well, what is it that we are trying to explain the origin of today? And Jim has highlighted 4 key classes of molecules. In addition to the classes of molecules, we now know that there are at least 2 main types of information processing systems at work inside the cell. There is DNA replication, the process by which DNA copies itself and makes more DNA. And there is something called protein synthesis, which we'll talk about in quite a bit in a minute. And that is the process by which the information in DNA directs the construction of the protein molecules, the protein machines that are necessary to keep cells alive, that do all the important jobs in the cell. So if we think about what life is today, we have these 4 classes of molecules that Jim was talking about: proteins, lipids, carbohydrates, RNA and DNA, the nucleic acids, but we also have, they are part of, or especially the nucleic acids and the proteins, part of complex information processing systems. A far cry from the protoplasmic goo of the 1870s. So the focus of my talk tonight is going to zero in on really 2 of these 4 classes of molecules: the DNA and the proteins that they produce. And indeed, the information processing systems in which these macromolecules or these biopolymers are embedded. Because in focusing on them, I think we can move from merely a critique of origin of life research to actually making a positive case for design. That in looking at the features of these critical molecules and the roles they play within these information processing systems, I think we are seeing crucial and definitive or distinctive indicators of the activity of a designing mind. So let's move forward with this. The story here is a great Cambridge story. It starts in 1952, 1953. Watson and Crick are here. They're wanting to do something, they say, about the DNA, something about the gene, and figure out the structure of the DNA molecule. In April of 1953, they published their famous article in Nature elucidating the structure of the molecule. 5 years later, Francis Crick, who was a code breaker in World War II, fittingly, I think, advances something which I think is an even more significant discovery than the discovery of the structure of the molecule itself. And that is that he realizes that the DNA, the chemical subunits along the spine of the DNA molecule, the so-called nucleotide bases, or the bases that Dr. Tour was talking about, that these bases are functioning like alphabetic characters in a written text or like digital characters in a section of machine code. In fact, the great physicist George Gamow, upon reading Crick's paper, said that the molecule and the subunits bore an uncanny resemblance to a digital bit string. This is information that's stored in a molecule for doing something. So what kind of information are we talking about? At the same time that Watson and Crick and the other great founders of the molecular biological revolution were elucidating all of these systems, there was also an information revolution going on. Claude Shannon, the great information theorist, developed a mathematical theory of information. And his basic idea was that information had to do with the reduction of uncertainty, and it was essentially something that could be measured mathematically in a probabilistic way. The more unlikely something is to happen, the more information is conveyed when it happens. So flipping a coin and it coming up heads gives me less information, 1 bit, than rolling a die when it comes up 3, because more uncertainty has been reduced and the more improbable event has occurred in the case of the die than in the case of the coin. In any case, this mathematical definition of information was very much in play in circulation during the 1950s. It was formulated, Shannon's theories were formulated in the late 40s, 1949, and Crick in particular was aware of this definition of information. Information was inversely related to probability. The more improbable something is, the more information is conveyed. But that definition of information, Shannon himself stipulated, could not capture the idea of semantics or meaning or function. That was something over and above. And so Crick was very quick to specify. He said, when he was asked, what type of information are we talking about inside DNA? He said, we're not talking about just Shannon information. We're not talking about something that's merely an improbable string of characters. We're talking about an improbable digital string that is sequenced in just the right way to perform a function. And so he says here, information here means the precise determination of sequence, either of the bases in the nucleic acid or the amino acid residues in the protein. We're talking about a different kind of information, not mere Shannon information, not mere improbability, sometimes also termed complexity, but we're talking about something that origin of life researchers later came to call specified complexity. The arrangement matters to the overall function of the system. Just as letters have to be arranged precisely to mean something in English, the A's, C's, G's, and T's in the DNA had to be arranged precisely to produce a functional protein. And we're a very mixed audience here. And if you've watched me on the internet, you know I use ridiculously simplistic visual aids, many of which I've stolen from my children. I use these in Cambridge with fear and trepidation because I know there are people from the molecular biological background, but it might be helpful just to get a little bit of a sense of what's going on here. So you've got the DNA molecule with these A's, C's, G's, and T's along the spine. And the cell takes that information, here's a nice depiction of it, and it directs the construction of these crucial protein molecules that are made of subunits called amino acids. There we are. So this is a little section of an amino acid chain. And if the A's, C's, G's, and T's on the DNA are arranged in the right sequence, they will, because of the molecular machinery at work in the cell, they will produce a sequence of amino acids that will fold into a very precise three-dimensional shape that will allow the amino acid chain to perform a function. So the amino acids form a chain, and then they have this, because of the interactions between the side chains and the amino acids, they will take on shapes. There'll be chemical interactions between them. If the shapes are right, then the chain can perform a function with a kind of hand-in-glove fit, okay? And so there's both a one-dimensional specificity of sequence in the amino acids and a three-dimensional specificity of form that allows these proteins to do specific jobs. And all of that derives from the prior specificity of the nucleotide bases along the DNA molecule. So it's not just Shannon information, it's specified and to a very high degree to perform a functional outcome. So just a few protein fold structures, these beautiful three-dimensional, highly irregular, but they're highly irregular in a way that matters to their function. This is a simple enzyme that breaks apart a two-part sugar, a disaccharide. And there's a little call-out window there that shows the way in which this disaccharide that looks like a little barbell nestles into the groove of the active site on that protein. And it's that type of hand-in-glove fit that allows proteins to perform their jobs. And we can think of proteins as kind of like the toolbox of the cell. They catalyze reactions at much faster rates than would happen in their absence. they form the structural parts of molecular machines, and they help process the information on the DNA. So they do all of these different jobs in virtue of their three-dimensional structures or shapes, or technical term, their conformations. So just like in the toolbox, the saw, the hammer, the spanner, they all have different shapes and therefore can do different jobs for the carpenter. So this is the sense of specificity that is produced by the DNA sequence. You get a one-dimensional specific array of amino acids that fold into a specific three-dimensional shape. So the term of art that was invented by the origin of life researcher Leslie Orgel was the idea of specified complexity. Complexity is effectively in the information sciences a synonym for improbability. So you've got something that's highly improbable, an improbable string of characters that are specifically arranged to do a job. That's what's got to be explained inside cells. And I've got a little animation now that actually shows the way that the information in the DNA directs the construction of the proteins. And this is the first of the two great information processing systems inside the cell that has to be explained because it is part of what makes up life. And let's just have a look at it. I think it's just amazing what's going on in our bodies right now as we speak.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is often transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
Because of you, millions of people have heard the gospel.
If you confess with your mouth Jesus is Lord and believe in your heart that he's risen from the dead, and you will be saved.
That's right.
That's the requirement.
We talk about science concepts which draw people in.
Take these nanomachines and have them drill into cells.
This would be a great way to kill cancer, right? We also talk about Jesus Christ, who's the best in everything.
My faith in Jesus Christ means more to me than anything.
If you could continue to give or give for the first time, we would certainly appreciate it. You can go to jesusandscience.org/donate. All U.S. donations are tax deductible. Thank you so much.
Okay, so that's the simple, homogeneous globule of undifferentiated protoplasm, no more. Okay, it's a completely new day in molecular biology and many leading origin of life researchers have recognized that of all the problems that Dr. Tuor has talked about, the most fundamental is the problem of the origin of the information, and I would add the information processing system that's at work inside the cells that make the proteins that make life possible. So what do we make of all this? Well, Richard Dawkins, who has continued to assert that all evidence of design has been explained by undirected processes such as natural selection, is himself expressed awe at this whole system. He says, the machine code of the genes is uncannily computer-like. Apart from differences in jargon, the pages of a molecular biology journal might be interchanged with those of a computer science journal. In other words, we have a striking appearance of design in living systems. But he has continued to assert that biology is the study of complicated things that merely give the appearance of having been designed for a purpose. So have these striking appearances of design, the information-bearing properties of DNA, the information-rich protein structures, the information processing systems at work inside cells been explained by any undirected material process? Well, in my work on the origin of life, both in my PhD years and in the subsequent years in which I was preparing the book Signature in the Cell, I realized that there are many, many origin of life models, but they all fall into three basic types of explanation. And one of the great molecular biologists laid this out early on. He worked closely with Crick. He was Jacques Monod. He wrote a famous book called Chance and Necessity. And he said, if you're going to be a scientist, you need to explain things either by random processes, stochastic processes of chance, or by processes, law-like processes of necessity. Or you might do like Darwin did and combine the two. And those are the categories of explanation we use. And in studying the origin of life and origin of life research, it turns out that these are the strategies that have been typically pursued. That some scientists have tried to explain the origin of the information in the DNA molecule or in RNA molecules as the result of pure random interactions between the subunits of those important information-bearing molecules. I'm not going to belabor any critique. I'm not even going to make a critique here. I'm just going to tell you the outcome of that approach within origin of life research, and that is that no one takes that seriously anymore. The amount of information that's present is so precise and so vast that even 3.8 billion years of cosmic history does not render the chance hypothesis remotely plausible. One origin of life researcher puts it this way. He said, blind chance is very limited. Low levels of cooperation, he, personifying chance, can produce exceedingly easily the equivalent of a few small words. But he becomes very quickly incompetent as the amount of organization or information increases. Very soon, indeed, long waiting periods and massive material resources become irrelevant. Now, Dr. Tuor has a way of demonstrating this chemically, that just allowing things to knock around will end up very quickly resulting in degradation. I have a way of demonstrating this mathematically and do that in Signature in the Cell, the math combined with the molecular biology. But for our present purposes and in interest of time, we're not really going to engage this because it's really not where the action is. Another approach to trying to explain the origin of information has been at different periods of time in origin of life research, because the fashions change, but it has been a very important approach. It's sometimes called self-organizational scenarios. It is effectively what Monod is talking about with necessity. It's the idea of invoking some sort of forces of physical chemical necessity to explain the precise sequencing of the amino acids in the proteins or the nucleotide bases, the information-bearing subunits in the DNA molecule. And this has turned out, it was a very popular approach in the 1970s. I had a debate last week where someone brought it up again, saying that law-like processes can produce complexity. Well, it turns out not to be true in the biological case, especially where this is relevant when we're talking about proteins in DNA. Scientists who first studied this, actually some of the scientists who first proposed this as a model, later realized that yes, there were some slight affinities between some amino acids and others that made them a bit more likely to bond to each other in some kind of a solution. but those slight affinities did not explain or correlate to the actual sequencing of amino acids you find in known proteins. So whatever produced proteins, it had nothing to do with chemicals knocking into each other and then binding by some sort of forces, law-like forces of attraction. That couldn't be the explanation. But there's an even deeper realization that came, and this was a realization that it's actually an amazing story, because the scientist who first proposed this idea of self-organizational systems was a man named Dean Kenyon. He wrote a book called Biochemical Predestination. He wasn't a Calvinist. He was talking about predestination in the sense of chemistry, of different constituents having differential attractions to one another. And he was challenged by one of his students about the viability of his model near the end of a spring term. And the student, I kid you not, was named Solomon Darwin. And he challenged Professor Kenyon, and he said, I don't think your idea of self-organization works when you're talking about the DNA, and that's the thing you most fundamentally need to explain. And so Kenyon went and he was given a book by a Swiss scientist who had first critiqued Kenyon's work, and he thought about it over the summer, and he came back and he said the 3 hardest words to say in the English language: I was wrong. And he realized that his model didn't work. And he later repudiated it publicly at a conference that I attended early in my career that was the event that first interested me in origin of life research. And it was a very, very poignant moment, because you had one of the most prominent people in the field at the time telling people, repudiating his own model and saying it doesn't work. Now, the exciting thing is that I think each one of us in this room can understand why it doesn't work. And in understanding that, we'll understand something really, I think, kind of profound about DNA. I wish I had a pointer, but I'm going to use my old-fashioned finger here. If you look at the DNA molecule, you see this is what's called a structural formula for DNA. And on each side of the molecule, you have the little P's for phosphates, and then the pentagons, which are the sugars. Is there such a thing? Oh, thank you, Alistair. Very good. You'll make me look high-tech. Okay, so you've got the sugars, phosphates, the backbone, and here again as well. And then along the interior of the molecule, you have these rectangular representations of the nucleotide bases. Those are the information-carrying subunits of the molecule. And it's in virtue of their precise sequence, again, that they convey information. Now, for the self-organizational model to work, there must be some kind of differential bonding affinities or forces of chemical attraction that can explain the sequencing, not of the sugar-phosphate backbone, not of this stuff, but of these guys, because they carry the information, right? And what you'll see is that these little sticks represent chemical bonds, and you'll see there's sticks between the sugars and the phosphates. There's sticks holding the nucleotide bases to the sugar-phosphate backbone, but there are no chemical bonds between the message-bearing characters in the genetic text, which means if there's no message-bearing if there's no bonds, there can be no differential bonding affinities. The chemistry, in other words, does not explain the sequencing. And to reinforce that point, it turns out that these bonds, by which the bases are stuck to the backboard, are identical kinds of bonds. They're called N-glycosidic bonds. So what that means is that any one of these bases, the A's, the G's, the C's, the T's, can stick to the backbone at any point. It's all chemically indeterminate. The chemistry allows for an indefinite arrangement of all this information-bearing text. Now, that was a lot of big words. Let me break it down even further. I have a little, not a backbone, not a chemical backbone, but a backboard, a magnetic backboard. And I've got some little letters with magnets. So there are, just as in the DNA molecule, there are forces of attraction that explain why the information-bearing characters stick to the backbone. In my visual illustration here, there are forces of attraction that explain why the letters stick to the backboard. But ask a question. Do those magnetic forces of attraction explain the precise sequencing here of my pandering message? Cambridge rocks. Are the magnetic forces of attraction responsible for the sequencing? Well, no, obviously, and I can prove it, right? Because I can rearrange all those things. The same forces are in play, but now I have a completely meaningless gibberish. Or I could perhaps spell something else. So all this is to illustrate what's going on here. That what we have is a true information carrier. This is not determined by physics and chemistry. The information is not determined by physics and chemistry, by underlying forces of attraction. Rather, it's independent of all that. And the great physical chemist, friend of Einstein's, Michael Polanyi, put it this way. He said, as the arrangement of a printed page is extraneous to the chemistry of the printed page, so is the base sequence in a DNA molecule extraneous to the chemical forces at work in the molecule. He used to say, somewhat cryptically, the information in the molecule must have an exogenous source, something outside the system of the physics and chemistry. And you can see this is very profound, because let's imagine for a moment that the A's, T's, G's, and C's were arranged as a result of some sort of kind of forces of physical attraction. Then every time you got an A, you would maybe have to get a G. And every time you got a G, you'd have to get another A. And then with that A, you'd have to get a G. And then the A would be followed by a G. And then you'd get not a message, but you'd get a mantra. You get a highly redundant system of sequence, a highly redundant sequence that conveys no information content. It could build, maybe you'd build one protein that way, but that would be all you'd ever build. And you wouldn't, because then you get highly repetitive sequences of amino acids that don't fold into those beautiful three-dimensional shapes. So it turns out that chemical indeterminacy is a necessary condition of the molecule storing, conveying useful information. And so what we've got here in the DNA is a true information-rich system. It's not an ordered system, it's a specified and complex system. And that's what needs to be explained, and it is not explained by underlying forces of physical, chemical necessity. All right, one more approach, a general approach, that's been tried in origin of life research. And that's the idea of some kind of prebiotic natural selection. And that would combine random variations of some kind with some sort of law-like process of selection, very Darwinian, as Darwin invoked, similar to the mechanism that Darwin invokes in biological evolution. But we're not talking about biological evolution here. We're talking about chemical evolution, the attempt to explain the origin of the first life from simpler, non-living chemicals. And so the idea is, well, maybe something like natural selection could be at work before you have life. But there's a problem here, and that is that natural selection in all living organisms depends on self-replication, the ability for molecules to copy themselves, and then essentially produce offspring that can compete in a competition for survival. But self-replication depends upon priorly existing sequence-specific DNA and protein molecules. Sequence-specific, another way of saying information-rich DNA and proteins. So in order to invoke natural selection, you need some process of self-replication, but to have self-replication, you need prior information, biological information in DNA and proteins. But what are we trying to explain? The origin of information in the first place, of biological information in the first place. So this is a formally question-begging argument. The great evolutionary biologist Theodosius Dobzhansky said that prebiotic natural selection was a contradiction in terms. Now, just to highlight that a little bit more, remember from my opening that there are two great information processing systems in the cell, or two main ones at least. One is the process of protein synthesis, which we saw in the animation. The other is the process of DNA replication. And in DNA replication, you have numerous exquisitely I'm just going to say, design proteins. They're just amazing. You have topoisomerases that unwind the supercoiling of the DNA molecule that allows it to be copied. You have helicases that separate the two strands. You have binding proteins that position the polymerases, the great molecular copy machines that come in and begin that process of copying the strands. It's an amazing system. There are animations of this up online, and I recommend you go look at it. It's just extraordinary. Every single engineering problem that you would encounter in the need to copy that information has an engineering solution in the form of a specific protein to do the job. But the big point, my main point here is simply that you have to have these proteins. And these proteins are produced by information, and so all of this is a precondition of biological self-replication. So you can't invoke natural selection, which depends on self-replication, if you're trying to explain the origin of information, because you need information to build the proteins that make replication and then natural selection possible. It's like the absent-minded philosopher of science who was walking home from his office one day and was thinking about something else and fell in a great big pit. And then he realized, well, there's no problem. I can get out of this pit. All I have to do is go home and get a ladder. So he got out of the pit and got a ladder and came back and jumped in and climbed out. There's something wrong with this explanation. It's begging the question of how he got out of the pit in the first place to get the ladder to get out of the pit. And this is the same sort of question-begging approach. Now, there are approaches that combine selection and random changes that are attempting to revive this notion of prebiotic natural selection at the level not of a single cell, but at the level of molecules, in particular RNA molecules. So if you've heard of this idea called the RNA world hypothesis or the RNA first model of the origin of life, that is an attempt to say, well, maybe you could have self-replication of individual molecules, RNA molecules, and once they copied themselves and began to compete, then you could get the process of natural selection going at a lower, less complex level. There's so much to say in critique of that, and we can talk a bit about it in the Q&A if you like. I'd love to, but just spoiler, and that is the bottom line here is that even if you got an RNA, the best we've been able to do in the lab is to get an RNA molecule that can copy about 10% of itself. But only then if the investigator precisely sequences the bases along the RNA chain so that you have an input of prior information to make even that limited self-replication possible. So again, you have before you get an inkling of self-replication, you have to have prior information. And so one of the great origin of life researchers of recent years, the Nobel laureate Christian Dedue, put it this way. He said, theories of prebiotic natural selection need information, which implies they have to presuppose what is to be explained in the first place. So where are we? Well, in my long study of origin of life research, I found that each one of these three basic approaches has turned out to be extremely problematic. Chance-based approaches turn out to be mathematically implausible in the extreme, chemically implausible for reasons that Dr. Tour alluded to. Chance plus necessity always presupposes prior unexplained information. The necessity approach ends up not being capable of explaining complexity and specificity, i.e. information. So each one of these seems to be deeply problematic for reasons that do not seem to be going away either anytime soon, or I would even argue fundamentally. I think there's deep theoretical problems with each of these approaches. So is there any other approach that, and I show the book just to show that there are many different models that exemplify each one of these approaches, and they all have one of two problems. Either they fail to explain the origin of information, or they presuppose massive amounts of unexplained information. But none of them provide an explanation for the origin of information in the first place. So question, is there anything of which we know that does produce and therefore explain the origin of information? Could there be a radical alternative that we're not considering? Well, I was first exposed to this idea when I met a scientist named Charles Thaxton at that same conference where Dean Kenyon announced that he was repudiating his own theory. Thaxton had recently written a book with two co-authors called The Mystery of Life's Origin, and it was a chapter-and-verse critique of origin of life research several decades ago. Nothing has really changed on that front, except in the epilogue to the book, Thaxton and his co-authors suggested that maybe we were barking up the wrong tree, that we were unnecessarily limiting ourselves to an ensemble of possible explanatory strategies that were inherently incapable of explaining the phenomenon in question. In other words, we're limiting ourselves to chance, necessity, the combination of the two strictly materialistic explanations when we're looking at something, namely functional or specified information, which we know from our ordinary experience is always a mind product. Is it possible that we are looking at a product of mind? Is it possible that we're actually looking at evidence of design? Now, after the conference where I met Dr. Thaxton and Professor Kenyon, I got deeply into this. And I started spending a lot of time talking to Thaxton. By the time I came here, I knew I wanted to do something on the origin of life. But I wasn't quite convinced of his idea of that the information necessarily required what he called an intelligent cause. And so I came with a burning question in the back of my mind, which is, could the design hypothesis, the idea of an intelligent agent being responsible for what we see, could that be formulated in a rigorous and positive scientific way? Could you formulate a positive scientific argument for the idea of intelligent design? And oddly, it was another Charles, not Charles Thaxton, but Charles Darwin, who first helped me make progress on this. Because he showed in his own writing that there were different types of science. There was a type of science that was concerned specifically with trying to reconstruct what happened in the remote past, a science that William Whewell called the paleontological sciences, the sciences of past causes, which I called in my PhD dissertation simply the historical sciences. And what Darwin showed was that in the historical sciences, we typically use a method for testing and evaluating competing hypotheses called the method of inference to the best explanation, or it's sometimes simply called the method of multiple competing hypotheses. Now, in my second year of the PhD, there was a young American professor who came to candidate for a job here in the Cambridge History and Philosophy Science Department. His name was Peter it's not Williams, friend of mine in the back, sorry, Peter Lipton. And Professor Lipton got the job, and he left behind, when he went back to finish his year in America, he left behind an unpublished manuscript. He gave it to me. It was called Inference to the Best Explanation. And he raised in this, he identified that, yes, this is how scientists are reasoning. They're comparing competing hypotheses, and they're electing that hypothesis which, if true, would best explain the evidence. But all that raised a question, Lipton said, and that is, what does it mean to be a best explanation? What is the criterion, or what are the criteria by which we determine when one explanation is better than another? And in his account of things, he identified the idea of causal adequacy or causal powers. If we can posit an explanation that invokes a cause which is known to produce the effect in question, that explanation is going to be better than hypotheses which posit entities that do not have demonstrated causal power with respect to the thing that needs to be explained. Now that was a lot of words, so let me break it down. Here, example. I live in the northwest corner of the United States, in the state called Washington. And when you go to eastern Washington, and you go and you look out on the wheat fields there in what's called the Palouse country, you can still find little patches of white powdery stuff out in the fields. And if you don't know what happened on May 18th, 1980, you would possibly use this method of multiple competing hypotheses. You'd look at the white powdery stuff and you'd say, well, could that have been produced by an earthquake? Maybe it was produced by a rainstorm. Maybe it was produced by a tornado. What else could it? Maybe it was produced by a volcanic eruption. Of those four hypotheses, which is best when we consider the need to provide a causally adequate explanation? That is to say, positing an entity or process which is known to produce the effect in question. Tornadoes? No, they stir stuff up. They don't leave patches. Rainstorms? No, they cause powdery stuff to leach into the ground. Earthquakes? Doesn't seem too likely. But what about the volcanic eruption? Well, yes, volcanoes produce white powdery stuff, and they often leave a lot of it behind for quite a long time. Now, it happens that with my somewhat contrived example, on May 18th, 1980, there was a massive volcanic eruption in our state, the famous Mount St. Helens eruption. So if you were there and you saw it, you wouldn't need to use the historical scientific method. But if you weren't there to see what happened, and you wanted to reconstruct what had happened, you would go and you would look at the data, and then you would compare the competing possible causal hypotheses, and you would elect that one which, if true, based on our knowledge of cause and effect, could explain the data. And so our knowledge of cause and effect is crucial to reconstructing the past. And this was the idea behind Charles Lyell's famous dictum of uniformitarianism. The present is the key to the past. This was a way of saying our present knowledge of cause and effect is the key to reconstructing the past. So here's a schema of how this method works. I've got an event that I need to explain. There's a number of possible causes I could posit. And if I'm fortunate, as I evaluate them, in the best case, it may be that there's only one known cause that produces the effect in question. If so, if I see the effect, I can retrodict, I can reason back to the activity of that cause. Now, when we're talking about the origin of information, well, I applied this method to the question of the origin of information. And I looked at hypotheses that were based on chance, variation, hypotheses based on natural law or necessity, hypotheses that combined the two. And none of them provided an adequate causal explanation for the origin of specified complexity or specified information. What about intelligent design? Could that be a causally adequate explanation? Well, as I was thinking about this, I came across this front piece in Charles Lyell's book. And Lyell had been a hugely important mentor for Darwin. In fact, if you walk across town and you go to Fitzwilliam Street, number 22 Fitzwilliam is where Darwin lived in 1836 and 37 when he came back from the Beagle voyage. And when he was there, he was talking to his mentor, Cambridge mentor Henslow, and to Lyell about what to make of all that he had found. In any case, on the front piece of Lyell's work on the principles of geology, he said this, he said, being an attempt to explain the former changes of the Earth's surface by reference to causes now in operation, causes that are known to produce the effect in question. This was Lipton's key insight, that the best explanations are ones that are uniquely causally adequate. Well, what does this tell us when we think about the origin of the information necessary to produce life? As I was pondering that, I had another aha moment. I was reading a book by an early pioneer in the application of the information sciences to analyzing molecular biology. His name was Henry Quasler. And he said, the creation of new information is habitually associated with conscious activity. Well, habitually associated got my attention because that's like uniform and repeated experience. This is the Lyellian dictum. But he also says there's only one known cause of the creation of information. It's conscious activity. And so I thought, well, is that right? Bill Gates has said that DNA is like a software program, but more advanced than any we've ever created. Richard Dawkins has said like machine code. Well, where does software come from? Where does a machine code come from? It comes from a mind, not from an undirected process. It comes from a programmer. And if we think about the origin of information in our uniform and repeated experience, whenever we see information of the specified kind that we've been talking about and we trace it back to its source, we always come to a mind, not a material process, whether we're talking about a hieroglyphic inscription or a paragraph in a book or a section or information that's embedded in a radio signal or coming across the internet. If you trace that information all the way back to its source, you always come to a mind, not an undirected material process. And so that suggests that the postulation of a prior mind, a mind prior to the origin of life, would be a causally adequate explanation for the origin of that information, whereas chance, necessity, and the combination of the two are not. In other words, using Darwin's own method of inference to the best explanation, the presence of information in the cell provides an empirical basis for inferring actual design, not just apparent design, when we're talking about the origin of information. Now, all this might seem, my examples might seem a little bit unscientific. Hieroglyphic inscriptions, okay, that's archaeology, paragraphs in books, radio signals, etc. All this is based on our ordinary experience. But remember, our uniform and repeated experience is the basis of all scientific reasoning. But you say, but I would like to see some sort of experiment that demonstrates that an intelligence is necessary to generate information. I refer you to Dr. Tour's lecture. Did you see that one slide where he put up the 7 steps in the recipe necessary to not get all the way to life, just to get to some biopolymers or some of the basic building blocks of those polymers? One of the great themes of Jim's lecture is that they cheat. That at every step along the way to make any progress in the direction of life-friendly chemistry, the origin of life researcher, in doing simulation experiments, is always constraining the outcomes, is always intervening, is always saying, I want this, not that. I want these reagents, not those. I don't want to allow this cross-reaction to happen, so I'm going to remove these molecules and leave those. Now, in a basic definition of information is that whenever you elect one thing and exclude another, you've imparted a bit of information. And there's a whole lot of excluding one thing and allowing something else in these simulation experiments. It's the only way you make even a modicum of progress. And so I ask the question, what is actually being simulated in origin of life research? In origin of life simulation experiments? Remember, the whole point of a simulation experiment is the present is the key to the past, the Lyellian dictum. And we're going to do an experiment that we hope will simulate something that might have happened in the past. And by watching what happens in our experiment, we can infer something about what must have happened in the past, or what might have happened in the past. But if in every single simulation experiment, there needs to be a guiding hand, there needs to be an input of information, an exclusion of some options and the election of others by an intelligent agent, what is being simulated? If not intelligence. If not intelligence. So I would argue that the simulation experiments themselves, though they have not gotten us anywhere close to life, are demonstrating the necessity of an informational input from a mind to even begin to make steps in that direction. And I think that provides further support for the design inference as the best explanation for the origin of the information we know is needed to produce the first life on Earth. So, conclusion is that prebiotic simulation experiments only increase biological relevance by investigator interference and intelligent inputs of information. So, what is being simulated, if not the need for intelligence? One of the great Cambridge scientists of all time, one of the great scientists of all time was, of course, James Clerk Maxwell. He founded the old Cavendish. You can go there and see the wonderful inscription on the door that he insisted was to be put there. And I came across this recently and found that he was actually very sympathetic to the kind of design reasoning that I'm explaining tonight. I'll have to decode this just a little bit, but this is a very, very profound quote. He says, the belief in design is a consequence of the laws of thought acting on the phenomena of perception. Now, the laws of thought include the idea that to explain something, you must invoke an adequate cause. And this was actually one of Newton's key principles of scientific reasoning. It was called the vera causa principle, and Lyell was repeating that. And so if you apply the vera causa principle to the question of the origin of specified complexity, which is part of our objects of perception, we have evidence of information-rich molecules in the cell. We apply the vera causa principle to that object of perception, and we should therefore conclude design, because we know in every other realm of experience where we see information-rich systems, it always requires an intelligent cause to produce the system. That's the vera causa. The true cause of information is, in fact, mind. Okay, let's wrap this up. I'm going to leave this to the discussion. If people would like to ask about whether or not this is a God of the gaps argument, I'd love to talk about that. And in the meantime, I think we'll leave things there. I've got a lovely little clip with Dawkins, but it's a very light. The clip, just in closing, is Dawkins being interviewed by Ben Stein in a movie in 2009. And I was at the time finishing Signature in the Cell. Ben Stein is saying, well, what are the chances that intelligent design could be part of the explanation for the origin of life? And Dawkins has just admitted that no one knows how life first began. So, you know what, Jim and I stand in no fear of contradiction tonight on that point. And Dawkins goes on to speculate, well, maybe there is a kind of signature of intelligence in the DNA, but it would have had to have arisen by, and then he punts to the space alien designer hypothesis, panspermia. But in any case, we'll leave that and turn it over to Alistair for a Q&A, and I thank you for your rapt attention.
Thank you for a wonderful pair of lectures, wonderful stuff. I hope this is relevant, but it seems to me that when we see mutations in cells, it leads to degradation and disease. But evolution requires precisely the opposite. Are there any known cases whereby we see positive mutations that lead to the benefit of the animal?
It's a big subject. There are mutations that lead to beneficial changes. But there are also, I think, increasingly, there's a recognition that there's a limitation to what mutation can achieve. The fundamental unit of biological form is the protein fold, the structure that allows proteins to perform different functions. And as there have been mutagenesis experiments showing that as mutations accumulate, over a very small number of mutations will degrade the stability, the thermodynamic stability of that protein fold. And the number that are necessary to degrade the fold are far smaller than the number that are necessary in what's called sequence space to generate a novel fold. So you can get variations within the structure of a given fold. Some of those variations can be favorable, but generating novel protein structure, the novel folds, seems to be beyond the reach of what mutation can do. And I'd refer you to the work in particular of an Israeli scientist, Dan Tawfik, alas, the late Dan Tawfik. He died in a tragic rock climbing accident. But he was here at the LMB and did a lot of this work when he went home to Israel. Our colleague Douglas Axe has also done a lot of this work both on the rarity of proteins among all the possible ways there are of arranging amino acids, called what's called sequence space, and also the isolation of proteins within that sequence space. So we don't deny, as a proponent of intelligent design, that mutations are real processes, that they can sometimes produce beneficial effects, but those the beneficial effects are limited by what we know about protein structure.
Hiya, you're right. There's a couple of things. I know there's, like repolarization, depolarization along the nerve. I know I was watching the BBC and there was something about, in nature, there's like ion waterfalls where electrons can be excited in like natural watercourses and then fall, and there's some sort of early place in history where you can see there's natural excitation and then of electrons and they fall back down again, and it mimics how things pass along the nerve membrane. But I think that's something where it's something in nature that somebody mentioned, and that might be similar to what happens in a human. And I guess if you come in from the evolution side, you're thinking of these, you'd have to fragment the theories that you're saying, and an evolutionist would say, well, let's look at how this starts, let's look at how this process starts. So what you're saying is it's irreducibly complex, I think, and you have to have intelligence. So I guess an evolutionist would have to look at one small section, and it's the link, like you say, between the DNA and the proteins, which part did that start at? I mean,
Doctor, what about the some mimesis of nature and living cells? Is there any sort of thing you would like to comment on that?
Well, yeah, we're talking about tonight mainly the origin of the first life, but your question and the one prior were addressing the plausibility of evolution, of the natural selection random mutation mechanism as a way of generating novel information once you already have life through the process of biological evolution. In my second book, Darwin's Doubt, I argued that there are limits to what mutation and selection can do, even in that context once you have life. But that really is a separate question from what we've been addressing tonight.
Hello, I'm Mina, long retired chemist. There seems to be an increasing consensus in the origin of life researchers that our laws of physics that we live and breathe are not conducive to reproduction, etc., on many levels. Therefore, there seems to be a growing search for different laws of physics that may have been extant 4 billion years ago. Do you think that that can have any legs?
No, and for the very reason I was discussing when I was talking about these self-organizational models, laws by definition describe repetitive patterns of order that can be captured either with a differential equation, captured meaning described with a differential equation or an algorithm. And these repetitive patterns, from an information standpoint, are highly redundant. They're not complex or improbable, they're highly redundant. And therefore, it's not the same kind of phenomenon that we find in the living cell with DNA or RNA or in proteins. Laws can do a good job of explaining what doesn't need to be explained in life. What needs to be explained in life is not repetition or symmetric order or repetitive order, but rather its specified complexity. And that, from our experience, we know, is only produced by intelligent agents.
I'll give them other laws. You want that? Fine. I mean, but then they're upset with the God of the gaps hypothesis. Then anything I can't explain, I can attribute to some other laws have been in operation. That doesn't do much for us as scientists. It's really a cop-out as a scientist to imply that there must have been other laws. It's like the origin of the universe. Everything came from nothing. And so the materialist will accept that big miracle, the big miracle. They will accept that and embrace that. And they may embrace that there were other laws in operation. But when you look at the forming of life, you have to, what they're doing is they're embracing many, many miracles again and again to imply what they have. So this goes right back to the same thing. If you want to mess around with science like this and start invoking one miracle after another, all we
change the definition of life, as you mentioned.
Yeah, change the definition of life, and we're pushing toward a model, say, of design, and that's forbidden by them. But they'll take all of these really extreme miracles one after another. So it seems to really be moving away from things.
Hi, thanks for the presentation. It seems to me that the only alternative is agnosticism, because both parties are suggesting infinite regress, whether it is information all the way to the back or design or intelligent mind all the way to the back. We still have to come to an assertion that one of these came into being suddenly with full complexity that can start life. So that seems to be a problem that both camps cannot solve easily. So how do you propose to do that? And my second is a scientific question. Where in this design argument does the designer stop messing with biology? Because the RNA that is being produced is agnostic. The ribosome is agnostic as long as there's a sequence that is at the start to start the translation process. Same thing with the transcription process. And then we have the existence of circular RNAs and long non-coding RNAs, which are also agnostic to the sequence that they are being produced at. So how do you reconcile these two problems?
Let me take the first one first and then ask for clarification on the second. I think the resolution of I don't think the two approaches are at all equivalent empirically. Our appeal to our own knowledge of cause and effect, our own knowledge of our uniform and repeated experience, says that we do know of a cause that generates large amounts of specified information, and that cause is mind. Material processes do not have that power. That has never been demonstrated. And so if we're to assess the competing approaches of the pure materialistic chemical evolutionary approach with the design inference is grounded in our uniform and repeated experience. Moreover, yes, it's true that you have a question of when all this started, but the idea that essentially every worldview or philosophical system has to posit something as the entity from which everything else came. And I think the materialistic worldview has long wanted to insist that that matter and energy are infinite in time and space, that they've always been here. And I think the cosmological and astrophysical developments of the 20th and 21st century are pointing inexorably back to a beginning point, such that the pure materialistic story, I think, is much less plausible. Something's got to be pre-existing. There must be something that's self-existing that explains everything else. And I think the idea of a pre-existing mind in light of the new cosmology is a much better explanation, a much better thing to posit as being pre-existing rather than matter. Matter seems to have had a beginning before which, or independent of which, there was no matter to do the causing. So I think both at the origin of life and the origin of the universe, the mind over matter view makes better sense based on our uniform and repeated experience and based on what we now know about the history of the cosmos. Your other question, can we take that one up separately? I'd like you to clarify what you mean by RNA being agnostic. Okay, thank you. But very astute question.
Hi, thanks for presenting and trying to make the case for Christian faith. But as you've reviewed and critiqued some of the origin of life scientists' work, many of those scientists actually will accept it. Their work can align with theism, even if they're not. But I'm struggling to understand what your proposal is, or for what is your biologically credible model for a 14 billion year old universe? Because I hear critique, but I don't see a concrete proposal.
What's my proposal for a 14 billion year old universe?
Yeah.
Absolutely nothing. I have no proposal for that. That's not a number that I've critiqued. That's a number that has been put forth by many scientists. So if you want to have a young Earth, I'll deal with that as well. You just have less time to deal with these issues. If you're going to talk about a pure materialistic model.
Yeah, my last slide had 4 pictures: chance, necessity, the combination of the two, intelligent design, but intelligent design with a number of little bars across it, indicating that it was in cognitive jail and couldn't be considered. There's a convention that arose not at the beginning of modern science, as we saw last night in Steve Snowbolen's essay, but really in the late 19th century. It's called methodological naturalism, and it says that if you're to be a scientist, you must limit yourself to strictly materialistic explanations for everything, including the origin of the universe, the origin of life, and the nature of our own consciousness. And that's part of what we're challenging, what I'm challenging as a proponent of intelligent design. I'm first of all arguing that the most causally adequate explanation, which is the criterion of interest when we're trying to reconstruct the past, is intelligent design. That's a positive hypothesis that has explanatory power. But I'm also challenging, and we're challenging, the idea of methodological naturalism, because it's the thing that has shaped our sensibilities that cause us to think, well, if I posit that a mind had a role in the generation of an information processing system, I've somehow violated the rules of science, or I don't have a positive proposal. That is a positive proposal. It's a causal explanation. And that's a legitimate part of science. In addition, once that explanation is posited, it leads to a whole different range of research interests and possible testable hypotheses. One of the lectures in this series at the very end is going to be about the junk DNA paradigm, which was a byproduct of it was a Darwinian interpretation of those sections of the DNA that don't code for proteins. And it was assumed by Crick and others when the non-coding regions of the DNA were discovered that those regions of the DNA must be the product of an accumulation of mutations over a long period of time. And that led to a discriminating prediction between the ID camp, the intelligent design theorists, and the neo-Darwinists. We thought, yes, mutations are a real process, but we wouldn't expect that 97% of the genome would be junk and that the signal would be dwarfed by the noise. So among our scientists, the prediction was made that as we learn more about those non-coding regions of the DNA, because we're dealing with a design system, we would expect that those non-coding regions would be importantly functional. And so the assumption or the perception or the inference to design generated a prediction that was testable. Post-ENCODE project, it's turned out that the ID prediction has been confirmed, and the neo-Darwinian assumption that all that non-coding region, those non-coding regions are junk, has been disconfirmed. So this shows the positive scientific program that flows out of the design inference. It's not just, oh, I'm going to say God did it, therefore there's nothing to do. Just like in the case of Newton, ID is a science starter, not a science stopper.
We're talking about smaller cells. Smallest bacteria have got thousands of proteins. E. coli has got more than 2,000. The smallest bacteria has got around 480 proteins. So that means there can't be anything smaller from a bacteria point of view. So the only explanation for that, for those 480 proteins to come to form the first or smallest bacteria, has to be intelligent design, isn't it? Because there's how can random mutations or natural selection select 480 interactive proteins to form the smallest bacteria?
There have been what are called minimal complexity experiments done on cells, and I think those experiments, if you remove certain proteins and see if you can get away with less, I think there have been, the conclusion is that between 250 and 400 proteins might be sufficient to sustain a minimally complex cell. So it may not, in your thinking about the origin of life, you might be able to set a little lower bar than a full bacterium with the number, the full complement that you mentioned, but you still have a huge problem. There's a huge informational requirement there, and it's on the base. It's not, and in the way you phrased it, I just want to clarify, we're not arguing for design. I'm not arguing for design simply because chance, necessity, and the combination of the two have been shown to be inadequate. It's also because we have positive knowledge of the causal powers of intelligence to produce the very kinds of things we see in living cells. Digitally encoded information, information processing systems, miniature nanomachinery, functionally integrated circuitry. The distinctive attributes of living systems are things we know from one and only one mode of experience, and that's with our experience of our own highly engineered systems. So I think it's a reasonable inference retrodictively to a mind prior to the origin of life.
So I don't generally use a design argument in that I'm limiting myself to the same thing that I hold my colleagues to. If you're going to use these instruments and these tools, I can take the same instruments and the same tools and show you why your own arguments don't work. And everything comes to a screaming halt when you use the very tools that they want to say we need to use. Yes, you don't have the informational code, but every step of the basic chemistry doesn't work. Every step of the basic chemistry just doesn't work. And so it falls apart at every turn, long before you get to thinking about a cell working. Just each of the components, the building blocks you can't make, the building blocks of the building blocks you can't make, and if given to you, you wouldn't know how to assemble them. So just even using the materialist's own tools, what they are proposing falls apart. And not just a little bit apart, it falls grossly apart.
Ladies and gentlemen, I want to thank you very much indeed for coming. Would you please join me in thanking our two speakers, Dr. James Tour and Dr. Stephen Meyer, please.
Thank you for joining me today. If you could give us a like, share, or podcast review, we would appreciate it. If you have any questions, you could send them to ask@jesusandscience.org, and we'll try to answer some of those questions in an upcoming video. And if you do not believe in the physical resurrection of Jesus Christ, and you want to hear about why I believe, send me an email to tour@drjamestour.org, and we'll get together by Zoom, and I'll share with you why I embrace the resurrection of Jesus Christ.